Okay, welcome back. Thank you very much for staying there. So we are going to move to the next session, probably the one that we are most proud. All previous researchers, they have been like, they are already people, leaders on the field. And now this session is devoted entirely for PhD students or during the first year of postdoc. I don't know if Paschalis, that is our next speaker, is already there.
There are two Paschalis. We are playing Waldo again. So
Okay.
I'm going to unmute both of you.
Okay.
Please, only the real Paschalis is allowed to share the screen.
Okay. I will present our next talk will be delivered by Paschalis Natsidis from the University. Okay, fantastic, Paschalis. Thank you very much. From the University College London in UK, working with Max Telford in Max Telford's lab. And he is going to present a talk titled Errors in Orthology Inference, a bug or a feature. It's all yours.
Yeah, thank you, Jesus and Rosa. And congrats for the event, by the way. Very nice. And the talk comes nicely after dear David's talk. I don't want to provide a negative feedback, but rather be constructive about that. So we are interested in reconstructing the early evolution of the animal kingdom using molecular data. And this means that we are both interested in the tree that relates to different animal phyla. And once we have a reliable tree, in using it to reconstruct the evolution of different animal characters, both phenotypic and genotypic. So the identification of orthologs is a fundamental step in this process.
Paschalis, sorry to interrupt. We don't see your screen. We just see it black.
Really?
Mm-hmm.
Give me a second, Wanda.
Yes.
Yeah.
When you select the option to share, put directly the program that you are going to use to reproduce the...
Yeah, I was using the Acrobat Reader, but I will try the PowerPoint now.
Okay.
Can you see something now?
Yes.
Yes. Perfect. Thank you.
Is it full screen?
Now, yes. Now, yes.
Cool. Let's go again. We are interested in reconstructing the early evolution of the animal kingdom using molecular data, as I said. We are interested both in the tree and in mapping characters on it. And molecular phylogenies almost always rely on the identification of orthologs, of aligned orthologs, to infer tree topologies and branch lengths. And for example, here is a recent phylogenomic tree that was built using aligned orthologous sequences. And such analysis greatly benefit from our ability to use genome sequencing to find all genes present in a species. This ability to examine complete gene sets has also allowed us to use orthologs and homologs in other ways too. First, this information can be used as a source of data for reconstructing the phylogenetic trees themselves. Here we see two recent publications that use the matrices of presence and absence of orthologs to infer the animal phylogeny. And these two trees are highly congruent with the phylogenomic tree I showed before. Second, orthologs can be used as characters that can be mapped onto the tree. As seen in these two publications from earlier this year, we can use changes in the genotype. We can link changes in the genotype, gene gains and gene losses, to changes in the phenotype and the evolution of new clades. It should be by now very clear, orthology, both of these types of analysis heavily rely on the accuracy of orthology inference. And as dear David said, the orthology is a central concept in evolutionary biology. As seen by the figure on the left, is a first and very important step in multiple analysis, apart from the two ones I already mentioned. However, there is still no one standard way of inferring orthology, and there are multiple methods and tools to do this task. On the right, there is a figure that compares true and false positive rates for a number of methods. And there has been a huge effort in benchmarking and improving this method. However, orthology inference remains a very challenging task. And the reason that it is still a challenging task is that genes and genomes evolve in a very complex manner. In the scope of this study, we focused on one of the factors that complicate orthology prediction, and this is the huge heterogeneity in evolutionary rates that is observed among different species, among different genes, and among different sites of the same gene. And we were interested in how these heterogeneities might result in errors in orthology prediction, and how will these errors affect subsequent analysis. And to investigate that, we performed a simple experiment using simulations focused on the animal kingdom, on the metazoa. And we decided to create artificial evolutionary scenarios where we will be able to better understand the source and the impact of those errors. We simulated sets of orthologs along the animal tree. And to do here, let me show you an example. Here is let's see that we look at these 12 hypothetical species and that they all share these 12 hypothetical orthologs. And this is a gene count matrix, and we see that all genes are present in all species as single copy orthologs, and no gene is missing from any species. Now, if we go and infer orthology in a scenario like that, since we know that most methods will not get it 100% right, we are more likely to end up with a picture like this. And while some of the orthogroups were correctly recovered, some others were split up during the inference and giving a false picture of smaller orthogroups and multiple gene absences, the zeros. So in order to do the simulations, we needed two things. We needed the guide tree, and we needed parameters of sequence evolution that will define the artificial evolutionary processes. And for the guide tree, we used the consensus of the current knowledge of the metazoan phylogeny. And for the branch lengths of the guide tree, we estimated them with a fixed topology using the C60 model in 93. For the parameters of sequence evolution, we collected 574 genes, ortholog genes, from these 57 species using OrthoFinder. And then we fed those genes into IQ-tree using a fixed topology to estimate sets of parameters such as branch and tree lengths, amino acid frequencies, and alpha parameters for rate variation among sites. And so we had the guide tree and the parameters, and we were ready to run the simulations, and we used the ALF software for that. In each simulation, we simulated the evolution of 5,000 genes along the guide tree, and to accommodate for the different rates that we see, in each simulation, we multiplied the tree length of the guide tree with a random multiplier between 0.2 and 5. So bigger multipliers, longer trees, meant that sequences will accumulate more mutations and thus emulate faster evolving genes. And for the alpha parameter, in each simulation, we used a random value from a distribution that was designed based on the empirical data I mentioned before. And since we were only interested in the effect of the rate heterogeneity in orthology prediction, we simulated without any gene gains, losses, or duplications to make our results more interpretable. And after we had the simulated sets of ortholog genes, we inferred orthology relationships among these sets. And for that, we used OrthoFinder with the default settings. And in each case, if the orthology prediction was correct, we would expect to recover exactly 5,000 orthogroups, and each orthogroup to have exactly 57 genes, one for each species. So any divergence from these numbers would be due to error in orthology prediction. Here is each dot represents one simulation, and we did 100 of them. Each one has a specific multiplier and alpha combination, and on the x-axis are the tree multipliers, and on the y-axis are the numbers of inferred orthogroups in each case. And the color gradient corresponds to the value of the alpha parameter. And we see that on the left hand of the graph, where are the smaller multipliers, the slower evolving simulations, that orthology prediction was very successful in recovering the correct number of orthogroups, the 5,000. However, moving to the right part of the graph, in the faster evolving simulations, we see that not only we start diverging from the expected 5,000, but also that the alpha parameter starts to play an important role in the result for a given specific multiplier. Looking at the mean size of the orthogroups, we see that this very quickly starts falling from the expected 57 towards much smaller sizes. So orthogroups start to get split up more and more as the underlying genes become faster and faster evolving. And of course, we wanted to see how these inevitable errors in orthology prediction, how they will affect downstream analysis, and particularly gene presence absence phylogenies. And of course, in a scenario like the one on the left, we would expect to recover a totally unresolved tree, a star tree, since there are no informative characters at all. But the question is what tree will we get in an occasion like the one on the right. So to examine that, we went on and built a tree based on gene presence absence matrices using RAXML. We built one phylogenetic tree for each of the 100 simulations. And we used the bin model of RAXML that describes the evolution of binary characters. And of course, as I said, in the simulation where we had almost no errors in orthology prediction, the gene presence absence matrix will be predominantly occupied by ones, and very few of the characters will be informative. And indeed, this is a tree that we got from a slow evolving simulation, and we see also from the color of the tips that the topology is essentially random. It is very different from the guide tree, the real tree. We wanted to quantify this difference, so we used the metric known as Robinson-Fouts distance. And the tree that I just showed you is somewhere up here on the top left with a fairly high RF distance. However, as we move towards the faster evolving simulations where we made more errors in orthology prediction, we see that these errors are not random, but the underlying gene presence absence trees get better and better every time. This is an example of a tree that we got from a simulation of a faster evolving setup. And we can see from the color of the clades that the different clades are all nicely recovered. Of course, the tree is not 100% accurate, but it is much better than the tree I showed before. I need to remind you here that we simulated without any gene gains and losses. And if the orthology prediction was correct in every case, we would expect to recover a star tree at all times. But we see that as we get more errors in orthology prediction, these errors are not random, but this tree, the phylogenetic signal that gave us this tree comes only from orthology errors. The tree, of course, is presence absence tree. So since the only force driving evolution in our simulations was sequence mutations, we wanted to see how the error that we get in orthology, how it is related to the branch lengths of the tree that depict the amount of these mutations. And for that, we counted all the pairwise patristic distances between all possible pairs of tips in the tree. And we also counted how many orthogroups each pair of taxa was inferred to have in common. For example, for these two members of the mollusk clade, we see that they have a relatively small patristic distance and that their number of common orthogroups is almost near the expected 5,000, very close to the truth. However, as we look to two members of the platyhelminth clade phylum, we see that they have a fairly larger patristic distance, and also that the number of common orthogroups inferred is much smaller than the expected 5,000. So we suspected one implication of that is that clades that have longer branches leading to them will have more clade-specific orthogroups falsely inferred. And this will have also implications on a subsequent gene gain and loss analysis, because these clades will have more clade-specific gains and clade-specific losses. This is in the paper that I mentioned in the beginning of my talk. The phyla of nematodes, platyhelminths and tardigrades, were found to have an increased number of gene losses compared to other phyla. But we also know that these phyla are known to have a long branch leading to them. And we wanted to see if these long branches somehow interfere with the gene gain loss inference. And to test that, we went back to our simulations, to our orthology results, in the simulated genes. And using a parsimony framework, a simple algorithm, we mapped gene gain and loss events onto the tree. And of course, even if we simulated without any gene gains and losses, we got multiple such events inferred based on orthology inference errors. And the three clades with the most falsely inferred gene losses were the tardigrades, the platyhelminths, and the nematodes. So this suggests that we should be very cautious when interpreting such results for such longer clades. And to conclude, we saw that errors in orthology that we get in orthology prediction correlate with lineages that are faster evolving and also genes and sites in a gene. We see that gene present subs and phylogenies are positively informed from these orthology errors, and that also that these errors may mislead and negatively inform gene gain loss inference, especially for longer lineages. And with that, I would like to thank, first of all, my funding body, the Ignite Network. Second, I would like to thank Max and Paschalia for their guidance in this project and in making this presentation. And also, I would like to thank, of course, Jesus and Rosa for inviting me and all the listeners. Thank you very much.
Thank you.
Thank you, Paschalis, for a very nice talk.
Thank you.
Okay, let's have a look at the questions. Let me get to Slack.
The first one is from James Fleming.
James Fleming is asking, I'm interested in how you think sequencing and assembly errors earlier in the sequence processing might compound the errors that exist in orthology inference.
Yeah, so we selected the most simplistic scenario, so we can only understand the impact of rate heterogeneities in orthology inference, but every complexity that you add, even if it's a biological complexity or a technical complexity, like the one James is mentioning, I don't think it will improve the result that we see, but probably only make it worse. But at which grade they will make it worse, it is to be investigated.
Steve Haddock has a question as well. How are the probabilities of gain and loss weighted? And are you using the guide tree as the truth or just as a starting point for simulation? In real life, that topology fluctuates dependent on inference methods.
Weighted gain and loss, I think, is Steve referring to the last bit with the parsimony framework? I think that it is 50-50 if it comes to a conflict. Otherwise, each site is equally weighted because it was a simple framework. Of course, there are much more complex ways to do that, and you can weigh the different sites of ones and zeros. And as a guide, for the guide tree, we use it as a starting point for simulation. And I don't know if it is the truth, as I don't know how it means the truth here, but in our analysis, yes, it was the truth. And you can use any truth you like. That's a cool thing.
Okay, no more questions for Paschalis. So thank you so much for your very inspiring talk.
If there are any more, I will answer in Slack. So
please do.
Fantastic. Thank you very much, Paschalis.
Bye-bye. Cheers.